So we now, from yesterday, we are done with the types of DNA. We discussed about various, because yesterday I was just teaching you very basics. It was not in hardcore details. So today we will focus more on the techniques also, and yeah, some of the models for the DNA replication and DNA repair system. So that should be our two focus now. Let's start. Yeah. So let's discuss the basics of DNA replication. So in this mechanism of DNA replication, it's a process of uh, in which your substances, enzymes, and proteins they all are used in order to replicate our DNA. So substances that we use while making the DNA replication happen are deoxyribonucleotides, enzymes like DNA helicase, DNA polymerase 3 to 1, RNA primase, and DNA ligase. And the protein that we use is the single stranded binding protein. So these are the all things that we are required for the DNA replications. I will repeat: deoxyribonucleotides, DNA helicase, DNA polymerase 3 to 1. Spelling is not right. Primase, DNA ligase, and protein that is SSB, single stranded binding proteins. So basically, this is your single double stranded template strands of DNA. So majorly what happened in your DNA replication, first of all, there's a replication fork is made. So because of that, you have two strands, leading strand and lagging strands. And the original strands remain there. So one end is from 5 prime to 3 prime end, other is a 3 prime to 5 prime end. So the one which will produce your DNA from the 5 prime to 3 prime end, this will be your leading strand with the help of DNA polymerase. On the other hand, which is running from anti-clockwise 5 prime to 3 prime end, here we also using DNA polymerase. While doing so, there will be some gaps, some broken parts of our new template, and these broken parts will be called as Okazaki fragments, and they could be fixed with the help of your DNA ligase. Then comes your, this is the simple way showing you how it's all starts. So here we have parent DNA with 5 prime to 3 prime and both had a double helix structure. And then you add your helicase enzyme which will unwind this double helix structure, and also your single stranded DNA binding proteins will make the templates available to RNA primase and DNA polymerase 3. And RNA primase will make primers for the production of your new template. Then, as I discussed, there will be leading strand and lagging strand. So the leading strand will start from 5 prime to 3 prime end with the help of DNA polymerase 3, starting producing corresponding template. Whereas in the lagging strand, DNA polymerase will 3 elongates the lagging strand, but sometimes it will be not completely full. It will start giving your Okazaki fragments, which will be filled with the help of DNA ligase enzyme. So let's go in more details how this is happening. So this is not just only one strand this is happening. This is like a zip is opening from in between. So off your bag sometimes your zip got broken, so it started to open from the center. So your DNA replication is like that. So it's get open from both side, left and side, right hand side. So overall directions of replication is in both side, left and right. The main thing is that one half of the end will be from the one template will be the lagging strand, and the other template would be the leading strand. Same on the another side, lagging strand and leading strand. Remember this thing. Yeah. So you have your helicase enzyme unzipping your DNA and your single stranded binding proteins producing primers, and in order to let DNA polymerase 3 to produce your new template. And in the lagging strand, your DNA polymerase 1, 3, they're all playing role primase. There are some Okazaki fragments are being there, and your DNA ligase is getting helping to fill it. So mechanism of replication starts at specific point of DNA molecule called point of ori. So at origin, DNA strand unwinds by breaking hydrogen bonds, and this takes place with the help of enzyme DNA helicase. So at the point separation it appears, it will make a Y shape and it is called as replication fork. So each old DNA strands act as a template for the synthesis of new strand. An SSB protein attaches to unwind strands and prevents rejoining. So the synthesis of new DNA strands and SSB proteins attached to the unwind strands prevent rejoining. And the synthesis of new DNA strands on the old strands takes place by an enzyme DNA polymerase 3, and it adds new nucleotides through complementary base pairing. Then DNA polymerase 3 always synthesizes new strands from 5 prime to 3 prime end direction, and the synthesis of new strands for the parent template strand heaving 3 to 5 prime end is continuous, and this strand is called leading strand. Another one which the broken part with the Okazaki fragments will be your lagging strand. And during this fragmentations, primase, RNA primase is the first synthesis RNA primer in that. And DNA polymerase here continues the synthesis of Okazaki fragments in the 5 prime to 3 prime end, and your RNA primer are replaced by the deoxyribonucleic acids with the help of DNA polymerase 1. And Okazaki fragments joins together with the help of an enzyme DNA ligase, and DNA polymerase 2 will once again checks the repairs occurs in a new strand. So that's what is happening. So here we can see this is the origin of replicase. The DNA strands unwinds at the replication of origin. So they start to open your double-stranded DNA like this, and hence the conclusion products will be the two molecules will be having two new circular DNA. That was very, I don't want to go in complex for the DNA complication this part. So I hope this DNA replication might be easy to understand and easy to get in. So just give me 5 minutes, students, and then we start with the DNA repair system. Yeah, start with this DNA repair system here. As I'm coming. Okay. Start with DNA. Basically, in DNA repair system, so we have discussed in quite detail about our nucleic acid, nucleotides, various type of DNA, how transcription, translation is done, how protein synthesis is taken care, how the DNA double helix structure is made up of. So we have discussed quite number of things till now. So now I want to take you that sometimes in our DNA system there could be sometime mismatching, nucleotide exchange, or mutation, site-directed mutagenesis, um, base pair mismatching. Also, many things could happen. Deamination could happen, methylation could happen, or your nucleotide could be removed. So, all these things could lead to drastic effects on the DNA, which will code later on harmful for the health or could create a bit problem to the body. So we will discuss about in this DNA, and if it is done, there is a automatic system in our body which will repair all these tragedies that are happening in our body in order to clear it out. So DNA repair. So damaged DNA, if it is in our body, it needs to be repaired. If the damage is passed on to the subsequent generations, then we call it as mutation, and it takes place in the germ cells, that is your eggs and sperms. If a damage is done to somatic cells other than the germ cells, then one individual will be affected, but not all. So damage, where does this damage takes place? It's happening because of there's some errors in the replication or some chemical agents acting on the DNA. UV lightning imparting energy into DNA molecule, and then spontaneous changes to the DNA. So these four reasons could be the reasons that damage could come from. So why need to repair DNA? So as of now, we have seen in the replication DNA polymerase do a great job, but not good enough. So there could be some errors of about 1 in 10 raised to power 7 nucleotides, which it does not correct. And another mechanisms exist to correct many of the errors left by the replication system, and most mistakes or damaged corrected by 99%, leaving just a few. So 1 in 10 is to over 9 and 9 errors are left. The mutations are permanent changes left in the DNA. So why repair DNA system? The repair of non-replication related to damage to the DNA must also be priority of the cell. So these defects also will prevent translation and duplication of the DNA. In them, cell will die. Again, any errors or changes to DNA become mutations, which are permanent changes left in the DNA. So, for example, sickle cell disease is a good example of devastating effects of tiny change to the DNA that is happening in the red blood cells, which are made up of hemoglobin. It is a large protein components, that is your two beta globin chains. A single base pair change and the substitution will cause a disease. For example, single strand of normal beta globin gene, that is your GTCCA, like this is a long chain. Then, but in the yellow portion, what you're seeing is here, the A adenine got mutated into thymine, and because of that, in the figure B, what you're seeing is a RBC cells in concave form, but in C they become sickle cell. So they cannot carry oxygen as good as it was in the B form. And this happened because of the change in the sequence of your first sequence, that is from A to T. There could be sometimes also spontaneous mutations, which involves thermal energy and happen due to the random molecular collisions between molecules and DNA in the cells. They cannot be prevented, but parts of the DNA molecules are stripped off and alterations are introduced. And there could be many outcomes of it. So, direct DNA damage. Some agents damage DNA directly. For example, chemicals and light. So, chemicals like alkylating agents, methyl and ethyl groups added to the DNA bases, and this type of damages can be repaired by direct reversal involving special enzymes, and they remove the offending atoms and restore the base. Then DNA damage, this is just a few types of damage, is repaired via simple reversible of the chemical change like UV light induced dimers, methylation of bases, ethylation of bases, large chemical groups added to the DNA. So these are the various most other damages required other system, but others are the most likely DNA damages that are happening. That is either ethylated, methylated, UV induced dimers, or large chemicals are added towards the DNA. So for example, in this figure, what we're seeing is here that random photons of ultraviolet UV they induce aberrant bonding between pyrimidines, that is your thymine and cysteine. So there's two thymines being here, and after the UV light interception, so they will make a bonding and will make a dimer, and this defect can be easily reversed back into the same without any dimer with the help of photo reactivation. So visible light energy is used to reverse this defect. If the UV light is coming to make our cell die, but then it could be reversed with the help of the light. So light is quite important, quite essential. And this is possible in the bacteria, yeast, protists, some plants, and some animals, but not in the humans. Like all the people who are living in the northern hemisphere above equator, so they might be having a lot of UV light there, not like in India. So they go for uh, sunshine bath, sun bath, and because of that, they had a this UV light also coming up, and they have a lot of mutations over the skins, and which cannot be reversed back. And the other forms of DNA damage, that is deamination, removal of your amino group of cytosine, and the base becomes uracil. Or deamination is also possible by adenine is removed and the base becomes hypoxanthine. Or guanine is removed and the base becomes hypoxanthine also. And depurination, in this, base is simply ripped off of the DNA molecule, leaving a gap like a missing tooth in them. So let's discuss at the molecular level one by one of each of them. So in depurination, in your DNA, your guanine is removed, so it becomes from cytosine to uracil. So your guanine is removed. In the depurination, your whole purine is removed, and then it is a depurinated sugar. In the deamination, your DNA is still there, but your cytosine uh, got changed, and it's changed into the uracil by the removal of the NH3 group, and oxygen is coming there. So this is deaminations. Further, DNA level of same two events ever as last happened. So once this is your deaminated C, so your cytosine with the G, it should be C, but it's not present. Yeah. So what will happen during the DNA replication? Two things could happen. One could be mutated, one could be unchanged. In the unchanged form, it will have the new strand with the C, but in another case, G has been changed to an A. So U will be, because of that, it will change into A. And in the depurination states, you don't have purine here, so there will be two cases. That is, your A and T nucleotides will be completely deleted, or in the unchanged version, A will come back. So these are the various possibilities are possible. So the cell has a big problem to overcome in this case. So how does it tell that which strand carried the correct informations? We think now. The cell has mechanism of identifying new strand synthesis by leaving nicks that DNA. So they have these small portions. These things are called as nicks, and they are the enzymes which can scan new regions for looking for the errors. So if your DNA mismatch is there, and this is the old strand, some gaps are available, newly DNA strand is there. So with the binding of DNA mismatch repair proteins, with the help of your DNA mismatch repair proteins, we will cover up this thing with cover up the nicks, and then remove all the newly synthesized DNA strand, and then replace with the help of your DNA polymerase and ligase. So that's the basic thing behind your all the DNA repair systems are happening. Whatever the problem is there, we completely remove the edited DNA pair system and add it with the new one. So in the correction mechanism, so first is direct reversal of the damage, which could be happen with the help of photoactivation. So the two thymines connected together by UV light could be removed with the help of photo reactivation. And excision repair, that is the one which has the removal of defective DNA. So there are three types you can repair them. That is your base excision method, nucleotide excision method, mismatch pair repair method. So in the base excision methods, the presence of uracil in the DNA is a great example of this type. The special enzymes replace just the defective base. So first we snip out the defective base, and second cut the DNA strand, third add fresh nucleotides, and fourth ligate the gap. And the nucleotide excision, same as previous, but in this it's recognized more varieties of damage and it's remove large segments of DNA in them, like 10 to 100 base pairs. In the mismatch pair repair, special enzymes scan the DNA for bulky alterations in the DNA double helix, and these are normally caused by mismatched bases, that is AG, AC, CT, and these are excised and DNA repaired. So basic mechanism is the same for all three types. So you remove the damaged region, resynthesize DNA, and then ligate. So this is your DNA, and this is the damage to the top strand happen. The first step would be to remove the damaged part. Then DNA polymerase will make new strand using the bottom strand as a template, and then DNA ligase will seal these nicks, these things that were broken. It will be repaired by the DNA ligase. So net result, repaired DNA. So as simple as that. Basic mechanism is the same for all three types, that is remove damaged region, resynthesize DNA, and then ligate them. So consider sunlight sunbathing or daily exposures. So impact of ozone depletion, impact on different skin tones, and environmental degradation is the reason also. So evolutions also act on the mutations. If we do not have mutations, then we would be all same. There will be no changes between each of us. So any change in environment would be deleterious to all members of the population equally. So there would be no evolution. But mutation does exist. It is supported by the comparisons of the related organisms. So this is showing whale and human comparisons of the genome, that it's almost similar, but we can see that whale size is so big and humans are so small. So that's it from the DNA repair system. I don't want to make it complicated for all of you, because I made this molecular biology class just for the basic students, so that everyone could understand it. There is no problem in understanding it. So in that sense, these workshops were created. So give me 5 minutes. We start with the another topic, and yeah. I think now you had learned quite a lot about the basics of molecular biology. I can teach you one more part about lac operon system and TRP operon system. Yeah, lac operon system. So basically, in the bacteria, genes are found in operons, and these operons are transcribed as a group and having a single promoter. And each operon consists of regulatory DNA sequence, which act as a binding sites for regulatory proteins that promote or inhibit transcription. And regulatory proteins that often bind to small molecules, which can make the protein active or inactive by changing its ability to bind DNA. So some operons are inducible, meaning that can be turned by the presence of particular small molecules. Another are repressible, meaning they are on the default by but can be turned off by small molecules. So how gene R expression is regulated? So there are various forms of gene regulation, that is mechanism for controlling which genes got expressed at what levels. However, a lot of gene regulations occurs at the level of transcription, and bacteria have specific regulatory molecules that control whether a particular gene will be transcribed into the messenger RNA. So in bacteria, genes are often found in operons. So here we can see that in bacteria related genes are often found in cluster on the chromosomes. So operons are common in bacteria, but they are rare in eukaryotic such as humans. So this is your bacterium, yeah, and this is your bacterial chromosome. And in this operon system promoter, where RNA polymerase binds with the gene 1, 2, and 3, and these genes will work together to do transcription with the help of messenger RNA. So you will have gene 1, 2, and 3, and then the translation will make protein 1, 2, and 3. So in this way, you're in bacteria, how the mainly your operon is working out. So let's see the anatomy of an operon systems. So operons aren't just made up of coding sequence of the genes. Instead, they contain regulatory DNA sequences that controls transcriptions of the operon, and typically these sequences are binding sites for regulatory proteins. So you have this RNA polymerase here with the promoter, and DNA where RNA polymer binds. So in the transcription part, you will have your gene 1, gene 2, and gene 3, and then they will produce your messenger RNA. Then most operons have other regulatory DNA sequences such as repressors that binds to the pieces of DNA of called operators. So this is the operator site where your repressor will come and doesn't let your RNA polymerase to be attached to the promoter. So because of that, no transcription will take place and no mRNA is made. And further, what we can see, there's activator site besides this promoter. If the activator comes, then the DNA is where activator binds and lots of transcriptions takes place. Lots of mRNA is made. Then anatomy of an operon further. So we can see that if the activator is off, then the no DNA binding But when activator is on, after having the addition of activator, so binding will takes place. So there are mainly two Operon system. One is LAC, one is TRP. TRP is an inducible Operon system used for the enzyme for the metabolism of sugar lactose. Whereas a TRP, it's a repressible Operon system which is used for the synthesis of amino acid tryptophan. Now let's check one by one each of these LAC Operon and TRP Operon system. So first is LAC Operon system. So majorly, if we see in this LAC Operon system, we having three genes, that is LAC Z, LAC Y, LAC A, and these genes are transcribed as single messenger RNA under the control of one promoter. This is your promoter where your RNA polymerase will attached, and you will have LAC Z, LAC Y, and LAC A. And these genes are transcribed as single messenger RNA under the control of one promoter. And genes in the LAC operon specify proteins that help the cell utilize lactose. So LAC Z encodes an enzyme that split lactose into monosaccharides that can be fed into the glycolysis. Similarly, LAC Y encodes for a membrane embedded transporter that helps bring lactose into the cell. So in addition to the three genes, the LAC operon also contains number of regulatory DNA sequences. These are regions of DNA to which particularly regulatory proteins can bind and controlling the transcription of the operon. So in the LAC operon system, here we can see cap site, where your promoter RNA polymerase binding site will attach, and this is the operator site where your repressor, that is which is blocking the RNA polymerase. So both are present. So we will see one by one how they functions. So promoter is the binding site for RNA polymerase enzyme that performs transcription, and the operator is a negatively regulatory site bound by the LAC repressor protein. And the cap binding site is a positive regulatory site that is bound by the catabolite activator, that is your cap. So let's take a closer look over the cap and their roles in the regulation of LAC operon system. So here what we can see in the first case, when there is no lactose, that is lactose is absent, so the LAC oppressor binds tightly to the operator. The repressor will come, it will attach to the operator, and it gets in the RNA polymerase way, preventing that transcription. So it doesn't let the transcription happen. Whereas with lactose, what will happen? Allolactose is a rearranged lactose part which will bind to the LAC repressor, and it doesn't let attach to the operator. So RNA polymerase can now transcribe the operon. So they will work as they should do. Further, if there is a cap, in the case of low glucose, when glucose levels are quite low, then CMP is produced, and this CMP will attach to the cap and would allow binding to the cap site, which will make your RNA polymerase attached to the promoter, and hence you will see high transcription. But once it is a high glucose, due to the high glucose, no CMP could be produced, and that's the reason cap cannot bind to the DNA. And without CMP, so transcription occurs only at low level. So even though RNA polymerase will be there, but it will be not as high transcription in the earlier case when there is a low glucose as compared to the high glucose. So what is happening in reality? Let's put it together. So glucose is present and lactose is absent. So in that case, your repressor will attach to the operator, and your cap will be not attached in the cap side. So you will have no transcription, nothing. But glucose is present and lactose is also present. So this will make little bit of transcription because allolactose will be produced, the rearranged version of lactose, and will make attach to the repressor and does not allow to attach to the operator. But what if glucose is absent and lactose is absent? So even though your CMP is attached to the cap site and letting attach to the cap site too, but RNA polymerase cannot go further because of the lactose absent, because your repressor will attach to the operator. And if your glucose is absent and lactose is present, so lactose will attach to the repressor, doesn't let to attach to the operator, but also your CMP will attach to the cap site and make very high transcription without any stoppage. So these were the four conditions. We summarize them here. Glucose present, lactose absent, cap binds, doesn't binds, repressor binds, so no transcription. Glucose present, lactose present, no cap bind, no repressor bind, low level transcription. No glucose, no lactose, cap binds, repressor binds, no transcription. No glucose with lactose with cap binds, no repressor, strong transcription. So I hope this lac operon system might be clear for all of you, and you will never forget this part. It was quite easy and easy to digest. And now we will go to the TRP part, trip operon system. The trip operon system, they are found in E. coli bacteria, which is a group of genes that encode biosynthetic enzyme for the amino acids tryptophan. So trip operon is expressed when tryptophan levels are low and repressed when they are high. So trip operon is regulated by the trip repressor when bound to tryptophan, and the trip repressor block expressions of the operon system. So let's check the TRP system. So instead of lac Z, XZY, XYZ, it has trip E, D, C, D, A, B, C, D, E, like in this reverse order. It also have promoter site. It's also producing mRNA and also producing proteins, and yeah, it's everything is similar. So it also has these operator site where your tryptophan is getting attached to it, to the TRP repressor, and RNA polymerase cannot be attached in the high tryptophan content, so no transcription. And trip repressor is inactive in the low tryptophan content. So that's the major difference in your TRP on and off system. So that's it. Nothing to think or discuss further in this topic. So I will share this trip lac operon system with you, which was quite essential. I want to switch to your little bit in your syllabus for today. So we are having still 45 minutes, and I should have incorporated. I have incorporated yesterday, I guess, for two techniques. One was molecular biology, was DNA extraction technique, and one was PCR technique. So third, I want to incorporate into this is the gel electrophoresis. So So let's discuss some blotting techniques that are happening in the um, molecular biology. So mainly there are around 7 blotting techniques available. That is southern blotting, northern blotting, western blotting, southern western blotting, northern western blotting, dot blotting, zoo blotting. Southern is done for to check DNA, northern for the RNA, western for the protein, southern western for the DNA and protein, and northern western for the protein and RNA. And we dot blotting is just a part of your southern blotting, and zoo blotting is just get to know about non-coding and coding regions in all species at once. So first is your southern blotting. So in this blotting technique, we detect target DNA fragments that have been size fractioned by gel electrophoresis. And this technique was invented in 1975 by E.M. Southern. And the principle in this technique was to exploit the property of radio-label probes with a single-stranded DNA. So if you want to detect the presence of specific sequence in our mixed DNA samples, then we will accordingly design the probe which will have complementary sequence to our target sequence. So what we do is first we get the DNA and then we blot this DNA with specific membrane. This is a nitrocellulose membrane. Then next, a probe consisting of purified single-stranded DNA corresponding specific gene is poured. Then any fragment that has nucleotide sequence complement to the probe will be sequenced with the probe. And then 32P, it will be radioactive, and then we check the complementary fragments. So this is how it look like. So you have your electrophoretic gel. Um, so all samples are being transferred, and then we transfer onto the nitrocellulose membrane. After transferring the nitrocellulose membrane, we put them into the radioactive labeled nucleic acids, and then we hybridize with the nucleic acid in order to identify our sample. So it's very useful to identify particular gene mapping in order to find out genetic pattern, mutation, deletion, duplication, gene arrangements, to determine the number of copies of particular DNA sequence. So it has parental testing. It has many use. So we will go through very nice technique that how southern blotting technique is done in the lab. So this video will be without sound, but it will have subtitles coming up with the detailed techniques that are happening in this. Let's start. So the same way what we have seen here, when you're doing your RNA, it's a particularly similar process, but we add RNAs also, because no, we don't, because in our body, in our skin, RNA is present, so this will kill the RNA. So we add diethyl pyrocarbonate (DPC), which inhibits the ribonuclease activity. Also, baking at temperature, high temperature destroys the ribonuclease activity. So all these things we taken care of. All processes are same. So let's just check its process, how it is done. Northern blotting. So let's continue to the Western blotting now. So basically, it's done with the help of SDS page, and the nitrocellulose membrane is also used, and we use primary and secondary antibody. So in this procedure, first all the samples of proteins are separated on the basis of a molecular mass on the SDS page or in two-dimensional electrophoresis. Then the electrophoresis moves to the proteins from the gel and onto the nitrocellulose membrane where proteins adhere to them. Then we detect a specific proteins and antibody to that protein must be available to it. So here we can see in this diagram. So the nitrocellulose membrane with attached proteins is there. Then we add to this nitrocellulose membrane your primary antibody, then the secondary antibody. Then towards the secondary antibody, your X-force, your chemiluminescence agents are added in order to visible your protein. So information obtained from Western blotting. Size of protein is possible, how much your expressed protein is, we can get to know about various diseases. So let's have a quick look over the detailed manner of your Western blotting.
In this video, you will learn how to perform a Western blot. A Western blot can be used to identify specific proteins in a sample and provide information about the protein size and relative abundance in the sample. First, fill a tray with blotting buffer. You will be using this buffer to equilibrate your gel prior to starting the Western blot. Next, remove the gel from the gel cassette using the opening key. Line up the arrows on the opening lever with the 4 arrows on the cassette to open the cassette. After trimming the top and bottom of the gel with a straight edge, equilibrate the gel in the tray with blotting buffer for 15 minutes on a rocking platform. Pre-soak fiber pads in blotting buffer so that they are thoroughly soaked. To make a blotting sandwich, obtain a container large enough to fit the gel holder and add enough blotting buffer until the container is filled approximately 1 centimeter deep. Place the gel holder cassette in the container with the black side down and immersed in the buffer, and the white side up and out of the buffer. Lay 1 fiber pad flat on the black plastic. Next, wet a piece of blotting paper and place it on top of the pad. Be careful to avoid any bubbles between the pad and the paper, and make certain the buffer covers the paper. Take the gel and carefully place it squarely onto the blotting paper. again being careful to avoid any bubbles between the gel and the blotting paper. Next, you will be applying a piece of nitrocellulose membrane. Remove the protective sheet from the membrane and wet the membrane with blotting buffer. Carefully place the membrane squarely on the gel. Avoid moving the membrane once placed on the gel, as proteins will begin to blot immediately. Using a roller, remove any air bubbles between the gel and the membrane. Place a second sheet of wet blotting paper on top of the nitrocellulose membrane. Place a second wet fiber pad on top of the blotting paper. Fold the clear plastic side of the gel holder over the sandwich and clamp it to the black plastic side by sliding over the white clip. This tight fit will squeeze the sandwich together. Insert the gel holder into the inner module. Make certain that the black side of the gel holder is next to the black side of the module. Place the inner module into the electrophoresis chamber. Add a frozen cooling unit and fill the chamber with blotting buffer to the level of the white clip on the gel holder. Place the lid on the electrophoresis chamber. Connect the electrical leads to the power supply, making sure the connections are correct, red to red and black to black. Turn on the power supply and run the blot at 20 volts. If a timer is available, set it for 2.5 hours. When the run is complete, turn off the power supply and disassemble the electrophoresis chamber and remove the inner module. Open the module and place it in a container filled with blotting buffer with the black side down. Starting with the first fiber pad, remove each layer until you reach the nitrocellulose membrane. As you remove the membrane, note that the proteins have been transferred from the gel to the membrane. Note that the kaleidoscope pre-stain standards have been transferred and can be seen on both sides of the membrane. You can also see that there are no longer any proteins on the gel. Immerse the membrane in 25 milliliters of blocking solution and incubate it for 15 minutes at room temperature on a rocking platform. Pour off the blocking solution. Add 10 milliliters of primary antibody. Incubate for 10 to 20 minutes on a rocking platform. The platform should be set at a faster setting to ensure constant coverage of the membrane. Pour off the primary antibody. Rinse the membrane quickly in 50 milliliters of wash buffer, and then discard the wash buffer. Add another 50 milliliters of wash buffer to the membrane, and let it wash for 3 minutes on the rocking platform at a medium speed setting. Discard the wash buffer. Add 10 milliliters of secondary antibody and incubate the membrane for 5 to 15 minutes on the rocking platform at a fast speed. Pour off the secondary antibody. Rinse the membrane quickly in 50 milliliters of wash buffer and discard the wash buffer. Add an additional 50 milliliters of wash buffer and wash the membrane for 3 minutes on the rocking platform on a medium speed setting. Discard the wash buffer. Add 10 milliliters of substrate. Incubate the membrane with the substrate for 10 to 30 minutes with either manual shaking or on a rocking platform. Watch for the color development. Once the colors have developed, rinse the membrane twice with distilled water and blot dry with a paper towel. Air dry for 3 to 60 minutes and then cover in plastic wrap for storage.
I hope you like all these sessions so far. Um, so we will make to an end soon with the dot blotting, and this is a southern western blotting, as we have discussed, is a combination of southern blotting and western blotting. It was described by Bowen and used to identify DNA binding proteins that specifically interacts with the chosen DNA fragments in the sequence specific manner. So sodium dodecyl sulfate SDS denaturing gel. Gel is then equilibrated in a SDS free buffer to remove detergents, and the proteins transferred by electroblotting to the immobilizing membrane. So during the transfer, proteins renature and hence DNA binding proteins may subsequently be detected on the membrane by the ability to bind radio-labeled DNA. The northern western blotting, it's a technique which is a combination of northern blotting and western blotting, and this technique is used for the identifications of protein RNA interaction, in which protein is run on a gel, blotted or probe with a labeled RNA of interest, and interactions are detected as hotspots on the protein. So dot blotting, it's a modified versions of western blotting, which is used for identification and analysis of protein of interest. So in this, it's differ from the traditional western blotting technique by not separating protein sample using electrophoresis, but sample proteins are instead spotted onto the membrane and hybridized with the antibody probe. And the zoo blotting, basically we having a non-coding sequence and coding sequence. So probes that are non-coding sequence, they just bind to themselves, to itself, that is your human, and these are the coding DNA, like for rat, cow, dog, human. So we can, when we run them, so we can identify that this particular area belongs to non-coding and coding of the human. So that's how the zoo blotting works. So thank you very much. That was the part for the blotting technique. Now maybe I can teach you one more technique, that is DNA chip technique. Yeah, because it's only DNA chip is a it's like chromatin. It's to and identify the chromatin immunoprecipitation. So this is also one of the important technique that we use nowadays for in the molecular biology. And these few slides are there. It will be not taking too long. So in chromatin immunoprecipitation technique, basically you take your cells and you find the cross-linked DNA in it, and then you put the antibody binding proteins to it, then immunoprecipitate it, and then wash the steps, and then after washing the steps, protein will be removed, antibody will be removed, and you will have on the cross-link reversal, and you quantify the DNA and quantitative PCR. So then you check for DNA and protein analysis later on. So what is chromatin immunoprecipitation? So not only do proteins interact with one another, they also interact with DNA. So chromatin immunoprecipitation is a technique that determines whether a protein of interest will interact with specific DNA sequence. And this technique is often used to study the sites on the DNA which bound to the particular transcription factors or histone proteins, and look at the precise genomic locations of various histones modifications. So how does CHIP works? So CHIP can be used to examine the presence of protein DNA interactions at steady state, or to quantify changes in interaction at specific phases of the cell cycles. And protein are associated with chromatins are temporarily cross-linked in the live cells. So we try to find out with the help of sonification in order to have fragments of 300 to 1000 base pairs. And the protein of interest, along with any associated DNA fragments, is immunoprecipitated from the cell's debris using specific antibody. And the cross-link is then reversed. DNA fragments are purified, and diluted DNA it could be assessed for the qRT-PCR using primer flanking and genomic locus of interest. Then DNA amplification is an indication of enrichment in binding of the protein of interest. So there are some modified form of chip technique. So first is chip on chip, that is genome-wide analysis of protein binding sites using microarray analysis of purified DNA fragments. And then ChipSec, which is a genome-wide analysis of protein binding sites using deep sequencing of purified DNA fragments. The native chip, which omits the cross-linking step, and we use mainly micrococcal nucleus digestions to cut DNA at histone linkers to examine the DNA target of the histone modifying proteins. Then chip exo, addition of an exonucleus digestion, step to obtain the increased resolutions of protein binding sites up to single base pairs. Then chia-PET, this is a chromatin interaction analysis by paired end-tag sequencing. This is a technique that combines the principle of chip with chromosome confirmation, 3C, to detect long-range chromatin interaction mediated via protein of interest. An iChip, this is indexing first chromatin immunoprecipitations, a high sensitive technique that reduces the number of cells required for chip experiments by initially barcoding total cellular chromatin. Then NChip, which is a technique which employs a CRISPR-Cas9 system to target specific genome regions. A guided RNA complementary to the desired genomic is expressed in the combination with the tag. An enzymatically inactive Cas9 protein is added to it. Then they are performed using the antibody against modified Cas9. And this could be very helpful to understand the cis-trans interaction between chromosomal loop events. There's a RIP chip, which is a similar technique in order to understand the protein RNA interaction. There are some limitations of CHIP. That is, as all with all molecular biology technique, CHIP is not without its set of limitations, such as cross-linking may additionally include proteins transiently interact with the DNA or DNA bonding proteins. Similar DNA interactions of proteins with short residence times may not be fully captured. Additionally, interacting proteins may mask the epitope of the protein of interest. Finally, the chip technique is extremely dependent on the quality and specificity of the antibody employed and may not discriminate between different DNA binding protein isoforms. Some factors need to take care, garbage in and out. So the chromatin generally in the chip can make difference in unpredictable, uninterpret- uninterpretable results. So in particular, sonication could make your chromatin break down. So we need to take care that it's associated from the DNA whose antibody epitope has been destroyed. Be sure keep chromatin on ice all times and limit pulses to under 30 seconds at a time to prevent proteins denaturations from overheating. And antibody validation. So these are done with the help of chip validation. Chip grade antibodies are recommended, but antibodies validated for immunoprecipitations or immunohistochemistry. Another consideration is the choice between your monoclonal antibody and polyclonal antibodies that you're using. Further, that has to taken care is a cell number that they should not be lot, they should not be less, they should be in optimum range. Cross-linking issue, they could cross-link to another chromatin which we are not looking for. That could happen. We should have appropriate controls like IgG control input, positive controls, negative controls like in all experiments. So that's something has to taken care while doing your chromatin immunoprecipitation. So let's see the last, oops, the battery is less. The last technique, how we do this in a YouTube, in the video format. So I will play a video now.
Come in closer, let me whisper something in your ear about Gypsy. Yeah, Stat Quest. Yeah. Hello, I'm Josh Starmer and welcome to StatQuest. Today we have a gentle introduction to ChIP-seq. Note, this StatQuest is based on the gentle introduction to RNA-seq, so watch that first unless you're already totally down with RNA-seq. Okay, we've got a bunch of cells. Inside each cell, there's a nucleus. And inside each nucleus, there are a bunch of chromosomes. Let's focus on the chromosomes. Specifically, let's focus on these chromosomes. Chromosomes are made out of chromatin. Chromatin is made out of DNA, plus histones, a type of protein, plus other proteins that we'll talk about some other day. They're not that important for understanding ChIP-seq. DNA wraps around the histones to package DNA, and the packaging can regulate gene transcription. Depending on how they are modified, histones can activate or repress genes. Lots more can be said about chromatin and how it's packaged, but that's for another day. Today we just need to know that chromatin is essentially DNA wrapped around histones. To simplify things, let's just use this big blue line to represent chromatin from here on out, but remembering that it's made of DNA and histone proteins. And let these big brown arrows represent genes. And let this green circle represent a histone that allows transcription. And let this red stop sign represent a histone that represses transcription. In a cell, all kinds of proteins can bind to DNA. This mustard-colored thing might promote gene transcription of this gene. And this pinkish-colored thing might repress this gene. And who knows what this green thing is doing? We can use ChIP-seq to find out. ChIP-seq stands for Chromatin Immunoprecipitation combined with high-throughput sequencing. It identifies the locations in the genome bound by proteins. That's the most important thing in this stat quest, so I'll say it again, in bold. ChIP-seq identifies the locations in the genome bound by proteins. For example, say like we wanted to identify all the regions in the genome bound by the green thing. The first thing we do is use formaldehyde, or something like it, to glue all the proteins bound to the DNA together with the DNA. This means that all of the DNA-bound proteins, including the ones we're not interested in, are glued to the DNA. The next thing we do is cut the DNA up into small, approximately 300 base pair, fragments. Then we isolate the protein we're interested in this case, we're interested in the green thing, using an antibody. The black star represents an antibody that is attached to a bead. Then we isolate the proteins bound by the antibody and wash everything else away. Then we reverse the formaldehyde glue by warming up everything. Then we isolate the DNA by washing away the proteins, including the histones. Now that we see how to isolate DNA that is bound by a particular protein, let's take a step back. So far, the example has focused on just these chromosomes. But the process, glue proteins to DNA, cut up DNA, bind proteins of interest with antibodies, isolate antibodies, unglue and wash away proteins, applies to all the chromosomes in the cell. And it is usually applied to a pool of 6 million cells, give or take a few. So we end up with a lot of DNA fragments from a lot of cells. Then, just like with RNA-seq, we prepare a sequencing library by adding sequencing adapters to both ends of the DNA fragments. Then, just like RNA-seq, we PCR amplify the library, check the library concentration, sequence, filter out garbage reads, and then align the high-quality reads to a genome. That is to say, if this is the genome, the first read might come from here, a location on chromosome 2. The second read might come from here, a location on chromosome 1. The third read might come from here, another position on chromosome 1, etc., etc., etc. Ultimately, we get a long list of genomic coordinates for all the reads, usually between 50 and 100 million reads. And we can use those reads to create a genome browser track. These are genes and chromosome positions in the mouse genome, MM10 to be exact. This is the track that we created for our ChIP-seq reads. A lot of reads map to this region, and relatively few reads map to these other regions. This track was made from a control experiment. The control track was made by taking some of the input chromatin from the original ChIP-seq experiment, and, without using an antibody to enrich for any particular protein, ungluing all the proteins and washing them off. Then sequencing, aligning, and converting into a track. In summary, the control track uses some of the same input chromatin for the ChIP-seq experiment, but doesn't try to enrich for any particular protein binding. We use the control track to verify that a high concentration of reads in the ChIP-seq track is due to a protein binding there, and not because a lot of reads map to a repetitive region. Statistically significant peaks are usually represented on genome tracks by rectangular bars. We could then compare peaks for the same protein in different cell types, like lung versus kidney. Or, if we didn't know the specific DNA sequence that this green thing bound to, we could guess that it is a motif found in all of the peaks. Here's a motif found within the peaks that indicate where the green thing bound, the large letters are more frequently associated with the green thing than the little letters. We can also try to determine the functional role of the green thing by looking at where it binds relative to the genes. Here, we see that the green thing binds near the start of a gene, so it might regulate that gene in some way. Anyway, those are some of the things you can do with ChIP-seq. In summary, ChIP-seq identifies the locations in the genome bound by proteins. Bam! Hooray! We made it to the end of another exciting stat quest. If you like this,
So thank you very much. I hope you had a good time here. Learn a lot of techniques within 2 days. Understand the basics of molecular biology, bit complex part also. So thank you very much, and please do give your comments on the this one on the Quora. So this will be very beneficial for us. So I will share it again. Give your comments. Yeah, thank you very much. See you tomorrow. Oh, bye bye. Wish you all the best for your future. Do the best and make your near and dear ones proud. God bless you.